All right, let's move on now to the methods and tools in cognitive neuroscience. First, EEG. So EEG, or electroencephalography, is a pretty old technique that's been around for about 100 years. But in the last decade, it's gained resurgence as the technology has improved and the analysis methods have become more advanced. EEG works by having an electrode on the scalp. The electrode is usually just a conductive metal placed on the head. As neurons under the electrode generate electrical activity when they are active, these signals sum and get detected by the electrode on the scalp. Signals are made up of oscillations of different frequency. One example oscillation is shown here. One thing to note is that EEG measures brain activity directly because it measures electrical signals which are used by the brain to communicate with one another. So that's EEG. We'll now move on to fMRI, which is probably what most of us have heard a lot about. In my 3 words, fMRI was one of the words that popped up in my mind when I think of cognitive neuroscience. What is fMRI? fMRI is functional magnetic resonance imaging. There are 2 important players in fMRI. One is blood. When a part of the brain is at rest, there is a certain mix of oxygenated blood, which are blood cells that carry oxygen, shown here. And there are also deoxygenated blood cells, which are cells that do not carry oxygen. The other player is neurons. So when neurons become active, as shown on the right, they require more oxygen to power the activity. The body responds by increasing the proportion of oxygenated blood to supply neurons with more oxygen. More than 30 years ago, almost exactly, it was discovered that oxygenated blood gives a very different signal in magnetic fields compared to deoxygenated blood. So fMRI is able to distinguish between brain areas that are more active from those that are less active. One important thing to note is that fMRI does not measure neuronal activity directly. It measures blood oxygen, which is a proxy for neural activity. That is why fMRI is also sometimes called blood oxygen signal imaging. So this plot gives a good overview of the properties of various imaging techniques. I just talked about EEG, which has very good temporal resolution, down to milliseconds, shown here, but poor spatial resolution. In contrast, fMRI has poor temporal resolution, slow, but it's got very good spatial resolution, meaning you can identify brain regions that are active within a few millimeters. Here you see other examples of imaging techniques and technologies and their resolution. EEG and fMRI have other weaknesses, including the need for subjects to limit their movements. One exciting new technique that is increasingly becoming more popular is called FNIRS, or functional near-infrared spectroscopy. FNIRS measures brain oxygen using infrared light and is much more tolerant of subject movement. This allows a more realistic study of cognition and learning in subjects that are moving and interacting. I bring this up because it's one of the more exciting developments in neurotechnology and will likely be increasingly used in learning research. My video is breaking up. Can everyone still hear me? Okay.
Yeah, we can still hear your audio. The video is a bit freeze, but the audio is still coming through.
All
so
right.
I think it's okay. It will come back again.
Okay, sure.
Yeah.
So now let's move on to comparing cognitive neuroscience to other learning sciences methodologies. I feel this part is quite important so that you can situate cognitive neuroscience within the broader schema of methods in the whole course that you'll learn in the next few months. So first, cognitive neuroscience is reductionist compared to socio-cultural methods. The premise is that the mind and behavior can be reduced to patterns of brain activity, while cognitive neuroscience acknowledges socio-cultural factors as important influences. The brain and how it controls the mind is the focus in cognitive neuroscience. Second, cognitive neuroscience is quantitative. It uses advanced statistical analysis, especially multivariate stats. You may have heard of ANOVA or ANCOVA, multiple regression, and so on. Cognitive neuroscience uses those statistics as well as many more. So if something is not quantifiable, cognitive neuroscience is unable to study it. Finally, the vast majority of cognitive neuroscience studies are done in the lab. There are very few studies of brain activity in real-world situations. Although with FNIR's technology that I mentioned earlier, there are more studies in recent years that go outside the lab. Nonetheless, the lack of the in situ nature of cognitive neuroscience raises questions about the generalizability of cognitive neuroscience. So despite its limitations, cognitive neuroscience is important for the learning sciences. This quote from psychologist Leslie Hart succinctly captures the importance of studying the brain for the field of education. Leslie Hart said that designing educational experiences without an understanding of the brain is like designing a glove without an understanding of the human hand. Cognitive neuroscience is a key discipline for us to understand the brain, so I personally feel it remains highly relevant for education. So we are done with this part on methods and tools in cognitive neuroscience. I think it's a good opportunity to discuss some questions and to hear thoughts from the class. These are some questions that I come up with, but feel free to bring up other issues or anything that might be unclear from my slides so far.